Dr. Ferdinand, and this is the first of a series of DAT tutorials in order for us to get you practicing and get you DAT ready. And actually, I should say DAT or OAT, depending on what you're studying for. But of course, most students, it will be for the DAT exam. You were looking at the brand new DAT Master Series in Biology, and it's because we're going to start by doing some practice questions in biology. So we'll be doing this every weekend on Sundays at 8 p.m. Eastern Standard Time, and which is not going to be a great time for the next few weeks because we're going to be up against the NBA playoffs and some other sporting events, but that's okay because studying for the DAT is more important than that, of course. So, so let me just give you a little background about what we're going to be doing. And then after that, I'll give you some time in order to complete the practice questions that we'll get done, and then we'll discuss the questions. We'll just go over a little bit of the background, and so that you can just have it like a study session. So of course, major spoiler alert, these questions are from the ADA. These are free DAT practice test questions. They contain real retired questions, as it's stated here. And in fact, the current ADA exam, so this is the exam that the ADA has available through Prometric. So it's a full-length DAT practice test. It is for 2022, and it has been adapted for 2022 because there are some new subjects. We'll talk about that in a second, but it also contains other subjects, many other subjects that the ADA has introduced since 2007. However, despite this, because I know some students, they think, oh my goodness, a test from 2007, that's going to be absolutely useless. But no, it's actually quite helpful for your to get the breadth of the exam. Perhaps the depth is not necessarily well reflected in 2007 because it's a relatively easy practice test compared to recent exams. But nonetheless, there's a lot of very helpful questions. How do we know that for sure? Because the current 2022 DAT practice test from the ADA is composed of some questions from 2007, some questions from 2009, plus a little magic number of questions that are unique to the new ADA exams. And that is what composes the current exam. And it is expected that this current exam will remain in place for many years unless there are new topics that are added in the future, which happens from time to time, and they may modify a certain number of questions based on those new topics. I put a little -E over here because traditionally the DAT had 5 multiple choice options. So you had A, B, C, D, or E, and that's the it was like this for decades. And although in this new exam they have removed E for most of the natural sciences questions. Of course, there's some DAT, PAT questions. There's no doubt. There's always 5. But for the natural sciences, they actually started weaning off. So some of the questions do have 5 choices, but most of them have 4 now. But the questions from 2007, almost all of them have 5 options. And even those questions that are used in 2022, usually one of those options are removed. Now, the relative difficulty of this exam compared to the real exam, of course. Well, first of all, it's a PDF, so you interact with it differently, obviously, than you would with a computer-based test, which the real exam is computer-based, held at test centers, Prometric test centers, and so it's a bit different. So there is that issue, but in terms of content, this is relatively easy. But just because it's relatively easy, it doesn't mean that it's not helpful. So, of course, it's helpful to go through the practice questions and to also dig into the background information, so that it's the chance that a retired question is going to be on your exam when you take the exam is almost impossible. But you do the practice questions and then you do the background information, because you should see practice questions, especially coming from the ADA, as a sign of the type of topics that would be important. So if you read around those topics, then you can be in a safe place for the real exam, as long as you do enough practice, of course. Not hundreds of questions, but thousands of questions. And speaking of hundreds of questions, so the natural sciences section has 100 questions, so 40 in biology. So that's the first 40 questions from the natural sciences section, 30 in general chemistry, and 30 in organic chemistry. So our plan is we're going to go through all of the questions. We'll just take it a little at a time, a few questions at a time, get you to try them. You'll be timed, and then we will, discuss it and go into a little bit of the background information. Okay, so now just in terms of strategy for these sections, normally, if you just go by using your dimensional analysis, which you'll be using for DAT QR, which we're not talking about today, but eventually we'll get to DAT QR. So using dimensional analysis, 90 minutes, over 100 questions. So that tells you how many minutes per question you'll have, which works out to 0.9, surprise. And so that works out to 54 seconds per question. So that's how much time that you have on average to complete the natural sciences section. However, biology tends to be, of course, you know it or you don't know it. So for the majority of biology questions, there's not a lot of time that you're going to be spending thinking about the questions. It's going to be more recognition of the content. But the ADA has evolved over the years to try to make the exam a little bit more problem solving. I know that sounds weird. Didn't I just get 100 questions? Aren't those problems? But the way it used to be is that almost all the questions, you just recognize the answer straight from memorization. But the ADA slowly been introducing more, some students might say a little bit alluding to the MCAT, a little bit more, certain levels of reasoning in order to answer the question. So that's been starting. Okay, so that's why a general rule of thumb that a lot of organizations would suggest is that you spend 30 seconds per question in biology. That means for the 40 questions in biology, you finish them in 20 minutes. So you really stick to it. Like you're continually looking at the clock and looking at the number of questions that you've completed and making sure that you remain on target. And then when we get to start talking about general chemistry and organic chemistry, when we get to those sections, we'll talk about strategies for timing and for time management, of course. So 30 seconds per question, that's usually the standard for biology. of course, it's going to depend on your academic background, the amount of studying that you've done, and so on. But just as a general rule, 30 seconds per question. Now, some students will, I mean, the 2022 ADA exam is a little bit expensive, okay, for what you get. But yeah, of course, if you can afford it, you do the free one, but also recognize that some of these questions are going to be in the 2022 exam. So keep that in mind, and we'll look at the percentage in just a moment, just before we start doing the practice questions. And there are some new topics. And so, you know, the ADA is going to reflect some of those new topics in the 2022 exam. And to be precise, they only announced in November that they were going to introduce these new topics, and then they introduced them in March 2022. So it's really recent that they introduced gene expression for biology, because we're doing biology today, genomics, epigenetics, and viruses and archaic bacteria. Now, the truth is that most students who studied for the DAT studied viruses anyway. They study viruses anyway, and they certainly would have explored archaebacteria in undergraduate, first year biology course, or not deeply, but enough to be able to answer a question on the DAT. So those ones, not so much. Gene expression, genomics, epigenetics, well, although those are covered, of course, in biology undergraduate courses, they certainly were not in the past DAT exams, and they are now appearing on real exams. So that is, partly part of the benefit. And I'll just show you one thing before we start doing practice questions. And so this is from the new 2022-2023 master series. And, at some point in the future, we'll talk about it. But right now, I just want to focus on something at the end of every chapter, which is called spoiler alert. So let me get to the end of a chapter and find a spoiler alert just to show you. Okay, so in the spoiler alert, it just gives you, first of all, the balance of the questions. So for DAT biology, it's 20% of the 2007 questions are in the 2022 exam, ADA DAT practice test. From the 2009, it's 45%. So that means about a third of the exam is brand spanking new practice questions. So you have to balance whether or not you think it's worth it. That's a personal choice. And then over here, it lays out the actual questions, what they are, of course, not giving all the multiple choice aspect of it, but giving you the idea of what the questions contain so that you have an understanding, and also what sections in the book refer to those questions. So it's important for you to keep in mind that the questions that we're doing today are real retired questions. So there's a true benefit for getting some of that practice. Okay, so what we'll do is we'll start off with the, providing you with the links. So the links to the free ADA practice test. And so I'm not here alone. I have some assistants, so they'll put the link there for you for the free ADA practice test. And then, normally it's 30 seconds per question. And in the future, that's what we'll be doing in the future, in the weeks to come. We'll be sticking to that kind of schedule, but we're just starting. And so you're going to get a bonus of 30 seconds per question. So we'll start with the first 4 questions, and actually we'll start with the first 5 questions. And so we'll start with the first 5 questions, and I'll just get myself a timer, and then you'll have 5 minutes to complete the first 5 questions from the PDF from the ADA, and then we will discuss those questions. Okay, so begin now. Okay, so that's all the bonus time you'll be getting. So let's take a look at the questions, starting with question number 1. Okay. Now, I know that for many of you, you will look at the question and think that it's very basic or very straightforward. But if you're just starting your preparation, which is it's fantastic if you're starting this time of the year. Usually students will take about a couple of months to complete their DAT studies and then take the exam during the summer, usually in order to have sufficient time to study and prepare. So if you are doing that, then you're doing a very good job. That's great. So now for the first question, it's important to keep in mind that the expression trophic, which you can see being used in all of these. Sorry, I just have to get the sheet. There we go. Which you can see being used in all your answer choices is referring to nutrition. So we're looking for some expression that's related to that nutrition. And the other issue is energy from light. So trophic is nutrition. Auto means that it is simple, as opposed to hetero, which is suggesting something other than what it is, like heterosexual, or it seems like a heterotroph. So it's something that's other than you. So heterotrophs are like humans or animals, that will eat, will find nutrition in plants or animals. So if that is the taking complex molecules and digesting them and breaking them down, if that is your thing, then you are a heterotroph. But if you're able to take carbon dioxide, I mean, come on, this is incredible. You can take carbon dioxide from the air. You can even take H2O from the air, and you can make a tree. You can make a bark. You can make very complex things from simple molecules. And if you are taking carbon dioxide and water and creating these complex things, then you know that we are talking about photosynthesis. And this is a difference between, a major difference between students who are studying for DAT or the MCAT, is that the DAT also includes plant biology, ecology, animal behavior, and some, I guess MCAT has psychiatry now, or psychology. So, but the DAT does have some topics, just in case for those of you who are using MCAT books to prepare, it doesn't cover the whole thing. And if you want to be sure you're staying on track, just Google the ADA DAT guide. There's a 2022 or whatever year you happen to be watching this, and go through their guide, and they have a syllabus in the guide that goes through all the topics that you need to know, and that includes plant biology for now. Okay, so the answer is A. It's autotrophic, because don't forget, it's not just taking simple molecules in order to make complex molecules, but it's using light, and photo is making us think about the photon, and the photon is an important part of the photosynthetic pathway, which occurs often occurs in a chloroplast. The other option for autotrophs are chemoautotrophs, and they take simple molecules, could be carbonates, could be nitrogen, but inorganic nitrogen and takes these simple molecules and then builds up the complexity. So those are chemoautotrophs, but they're not using light. And that's the key in the question stem. Okay, I think we beat that up enough. So let's move on to question number 2. Of course, if anybody has any questions at any point, please feel free to ask. So question number 2 is about fermentation. And in fact, the next 4 questions are quite related, of course, because it's metabolism. So there's question number 2, which is fermentation. Number 3 is respiration. Number 4, it's the hydrolysis of a complex molecule to be discussed. And then number 5 is an organelle that is very important, and we've already mentioned that organelle. Okay, so for number 2, though, this is a question with regards to fermentation. And so I think that most high school students would correctly answer this question. But nonetheless, let's take a quick peek of the process itself. And just get this out here. Let's get to and here we are, the summary of the production of ATP. Okay, so we have these 2 classic pathways, anaerobic, which is mentioned in this question, in one of the answer choices to this question, and aerobic, which is also mentioned. So, of course, anaerobic, because aerobic suggests with oxygen. So anaerobic, of course, when you're doing aerobics, you consume a significant amount of oxygen. And so that's aerobic. That's the aerobic pathway. And anaerobic would mean the opposite of aerobic. And so we have the pathway without oxygen. This occurs in the cytosol of eukaryotic cells. And here, glucose is broken down, glycolysis. Glyco refers to the glucose, and lysis is commonly used in biology to mean something broke apart. Hydrolytic, pathway, or the lytic life cycle of a virus. Something is going to break apart, and that's why that's where the word lysis comes. So glucose, a 6-carbon compound, broken apart into 2. Into pyruvate, which is a 3-carbon compound. So, and it goes through the pathway of glycolysis with trioses being produced. And finally, lactate. So you will see this written as lactate or lactic acid. And this builds up in the anaerobic pathway for somebody who's, running very quickly, and starts building up the lactic acid. And so they will start breathing more, and they'll have some oxygen debt because the body wants to oxidize up, or at least oxidize back up to the Krebs cycle in order to get more ATP, greater efficiency from the substrate. Okay, so we see that we get 2 ATP that's formed per molecule of glucose through the anaerobic pathway. And this is the final product. However, if there is sufficient oxidation oxygen available, then we can go up to the mitochondria, because pyruvate acetyl coenzyme A through into the mitochondria, and then we can have a slower system of biochemical events, but it is far more efficient in terms of the production of ATP. And here we have the waste products of the oxidation of glucose. So it's easy to imagine carbon dioxide as a waste product, but it's let's say for most students who are just learning biology, the idea that water is a waste product is for some students is counterintuitive. And it's not just that it's a waste product, but we will see in another question, it is actually produced through the reduction of oxygen. So we'll be looking at that shortly. But the main thing for this question is to clearly show that that the pathway, of course, that is more efficient for the production of ATP is the aerobic pathway. And now I know some of you, some of you will look at that 36 ATP and cringe because most professors feel uncomfortable seeing that, but there are some that still sort of use that figure sort of as a legacy almost. But of course, it's not 36 ATP per molecule. It's less than that. The system is just not that efficient. Although, there are certain numbers that you can learn during your preparation, about NADH2 coming up with 3 ATP, FADH 2 ATP, GTP producing approximately 1 ATP per GTP, and so on. But in the end, overall, these are approximations, and the ideal is not achieved. So for the question regarding fermentation, it's easy to see that, for example, answer choice A, pyruvic acid as an end product, actually it's an intermediate, and the end product is a lactic acid or lactate and 2 ATP. So answer choice A is incorrect. But B says yield less energy per mole of glucose in aerobic respiration. Well, clearly it yield less energy per molecule. So that is clearly the correct answer. And other answer choices yield energy in the form of ATP as it passed down the respiratory train. Sorry about that. Seems that everything technical is back on track. So, okay. So I'm not quite sure at which point it became unclear, but I just went through the various answer choices to show that answer choice A produces pyruvic acid as a end product is incorrect. It's producing lactic acid as an end product or lactate. And so it's just an intermediate in terms of pyruvate or pyruvic acid, but answer choice B yields less energy per mole. Okay, that's clear. Come on, there's 2 ATP per mole of glucose and 36 ATP per mole of glucose here. Now, 36 will make a lot of people uncomfortable. Some professors will not accept that. Completely understandable because it's not even the case, but this is a legacy issue. It's the first confident calculation, but the system is just not that efficient. But students still tend to remember that NADH2 through the electron transport chain can deliver 3 ATP per molecule. FADH plus H can deliver 2 ATP per molecule. GTP can deliver ATP, 1 per 1. And so those are just, generalities. But you see that the question is constructed such that you don't memorize the number 36. Okay, and occurs only in the presence of oxygen? Clearly not. It occurs, it's an anaerobic process, prevents glycolysis from occurring. Fermentation prevents glycolysis from occurring, but glycolysis, the breaking down of glucose, lysis of glucose, occurs as a matter of fact in the process of fermentation, and it converts ethanol to glucose. That is clearly incorrect. It's breaking down glucose. Okay, so that's, I believe that's enough for question number 2. And unless there's any questions, and so we'll move into question number 3. Oh, I also put here cross references if anybody happens to own the Master Series book, then details are provided for the first question in biology, section 2.2, that means chapter 2, and 16.6.3, that means chapter 16. And for respiration, metabolism, biology 4.4 to 4.10, and that's going to be the same for question number 3. Why? Because question number 3 is looking at what does oxygen do in respiration. So let's have a look. What does oxygen do in respiration? So we're turning away from fermentation and taking a little look at what happens in the mitochondria. So here we have the part of the electron transport chain, and we can see, within this process, there is oxidative phosphorylation. So first of all, let's look at how we have a substrate. So the substrate is, the basic chemical that is being used to transfer reducing equivalents. Sometimes substrates are used to transfer phosphorus or phosphate onto ADP to make ATP. That's substrate level phosphorylation, but this is oxidative phosphorylation. And in this process, we have something that is reduced. Okay, so this expression to reduce something or to oxidize something, you're going to use this expression differently in organic chemistry as you would in let's say biochemistry, if you will. Of course, this is clearly a component of biology. So in organic chemistry, when you're thinking about reduction, oxidation, you're usually thinking about oxidation means gaining oxygen or losing hydrogen. Okay, so just, we're going to go over that, in a few weeks when we get to that. But oxidation is gaining oxygen, so gaining oxygen content or losing hydrogen. And reduction is the opposite. So reduction is gaining hydrogen or losing oxygen. Okay, when you get to general chemistry, you look at it just differently. Some people have Leo Deline says GER, or Leo is a jerk, and Leo is a jerk stands for loss electron oxidation. So loss electron oxidation is anode, gain electron reduction, cathode. So we have the substrate here, which is reduced because it has that, it has the hydrogen. And when you talk about hydrogen in this context, please don't confuse this with a proton or acid-base chemistry, because acid-base chemistry is different. So there's 2 levels here. This is reduced, not an acid, because it has hydrogen with its electrons. If it was H plus and it was only releasing H plus, that's different. But you see how this, that is an acid. Okay, and when I say that's different, that's an acid. But this has H plus, but it also has hydrogen with electrons. And this is being passed along this respiratory chain, respiratory chain, or the electron transport system. So we have H plus that's being pumped out, and of course that hydrogen is very important, as there is a proton pump that is able to concentrate the H plus and pass through a receptor in the mitochondrion that is able to generate ATP. So by concentrating it on one side and then releasing to the other side. And please don't ever make that mental error of when there's high hydrogen concentration, it means low pH. Don't make that opposite thing, because sometimes they'll ask on what side of the membrane would you expect the pH to be higher? So you expect it to be lower on the side of the membrane that's concentrating the hydrogen. Okay, so now the electrons through H are being passed through ADH, flavoprotein, and the cytochromes, B, C, A in that order. And of course, the cytochromes use iron for oxidation reduction, so for redox. And you see the protons that are formed through this process, they combine with oxygen in the final step to produce water, which is the waste product of oxidation, aerobic oxidation or respiration. Okay, so then for question number 3, which asks, in respiration, what does oxygen do? We saw that oxygen acts as an acceptor for electrons and protons. Okay, so it's not just oxygen having, oxygen needs to have the electrons and the protons in order to have the final reduction that occurs. So it's the reduction of oxygen, because it's not obviously adding oxygen content to itself, it's adding hydrogen content to itself, and that is what we said is reduction. Okay, so the answer to 3 is indeed D. And now let's look at question number 4. So just to give you an idea, questions 1, 2, 3, the old DAT used to be almost all like that. There wouldn't be, so much of a preamble as there is with question number 4. So question number 4 has a preamble to set things up to at least have one extra level of logic or reasoning, but it still produces a very straightforward question. So an enzyme is added, aqueous solution. So we've got ATP. So we have energy. We have an enzyme, we have DNA, we have albumin, misspelt, a little typo there. I think that's some ancient way to spell albumin, ending with it. Albumin, so to speak. So anyway, fat and glycogen. So incubated, and now we have the mix plus glucose. So the question is, what in the original mixture produced glucose? Well, albumin is a very famous blood protein, very important in terms of maintaining oncotic pressure within the bloodstream. So that's the pressure that is due to various molecules, particularly proteins. And if the level of those proteins in the bloodstream is low, then fluid, this is going to affect the movement of fluid, and usually fluid will then move out of the bloodstream into the third space, and that's called edema. So it'll move into the third space, also called the interstitial spaces. So fluid in the body can be in cells, it could be in the blood vessels, so intracellular, in the blood vessels, intravascular. And then there's that third space, which is extracellular, and that's concerned with the lymphatic system, is very concerned with the third space. And then there's fat, as an option. So we would think that would be broken down into fatty acids. It's certainly not glucose, and albumin would be broken down into amino acids, because those are the monomers that create these polymers. And a glycogen being another polymer, which is concentrated in the liver, in hepatic cells in the liver, as a storage of glucose. Just like glycolysis refers to the breaking down of glucose, we have glycogen, which generates a long chain of glucose molecules, or it is itself a long, complex chain of and branching of glucose molecules, which is stored to be used, of course, in the liver, but also, of course, in muscle. In many cells, but these are the cells that are most concentrated. Muscle cells, myocytes, liver cells, hepatocytes. And then there's ATP. We've already, ATP can be digested, and you have inorganic phosphate, and then you have DNA, the nucleotides or nucleoside phosphates. So none of those are referring to glucose. So glucose is coming from glycogen, and yes, that is not the most difficult question that you'll see. But don't forget, 20% of the questions here will end up in the 2022 exam. So that's a spoiler alert for you. And so that's answer choice. The answer is C for 4, and for background information, go to 4.1 or 6.3.4. Okay, and now let's look at what cellular organelles would you expect to be absent from fungi. So fungi, of course, are very famous eukaryotes. So they do have, of course, there's prokaryotes that are pre-nucleus organisms, and then there are eukaryotes, which are true nucleus organisms. And prokaryotes have no membrane-bound intracellular organelles. So no membrane-bound intracellular organelles. So that means, do prokaryotes have lysosomes? No. Do prokaryotes have mitochondria? No. In fact, the prokaryote is approximately the size of a mitochondrion, and that's why, and mitochondria is the only place outside of the nucleus that has DNA. So that's why it is felt that there may have been a moment in the history of life where prokaryotes, did a little mutualism with eukaryotes, and then finally just got used by us, something like that. And so when we're talking about fungi, though, now we're talking about eukaryotes. So these have membrane-bound organelles. Mitochondria, of course. Lysosomes, of course. Ribosomes, that's an unfortunate name, a ribosome, because you hear lysosome and it's a membrane-bound organelle. It's sometimes called, it's the organelle that can digest molecules in the cells. It can break things apart. It can break even old organelles apart into their substituents so that you can reuse the beneficial molecules that are broken down into those substituents. However, I'm saying it's an unfortunate name because many students have made the mistake of hearing ribosomes and asking about a prokaryote and seeing lysosomes next to ribosomes and not recognizing that a lysosome is a membrane-bound organelle and therefore cannot be in a prokaryote, but a ribosome is not membrane bound. It is a complex of ribosomal RNA and protein. And of course, there are ribosomes in prokaryotes as there are in eukaryotes, different types, different weight, but they occur in both because both produce proteins, and ribosomes are provide the environment for protein synthesis, whether it be free ribosomes in the cytosol or ribosomes attached to a endoplasmic reticulum membrane in eukaryotes, and that creates proteins that are destined for the membrane of the cell or for exocytosis and use in the body for various reasons. Could be hormones, could be antibodies, could be various reasons. And Golgi bodies, sometimes the ADA updates that expression to the Golgi apparatus, perfectly legitimate expression from the 2007 exam. And of course, when you need to do any exporting of proteins that have been produced, synthesized in the cell, yes, wonderful. You use the Golgi apparatus, the export department of the cell, and that could send molecules to the cell membrane or for exocytosis. And then chloroplasts. Right. Yeah. Photosynthesis, not a favorite thing for fungi. They tend to break down molecules that are from their environment. For example, they can break down complex molecules from their environment. But using light in order to generate complex molecules is not the domain of eukaryotes. It's not the domain of fungi. It is the domain of eukaryotes and prokaryotes. But for eukaryotes, tends to be plants, and plant biology is part of DAT. Okay, so that's E and that's Bio 2.3. And despite the technical hiccups, it looks like we're pretty much on time for a 1 hour webinar. So we did 5 questions today. Next time we may spend a little less time per question, because I am hoping that those of you who may watch the video or who are here right now might actually try some of the questions prior to the next week's webinar. But it's okay. If you do not try, you'll have some time during the webinar, but less than 1 minute per question for biology. You may get more for general chemistry, and you may get 1 minute for organic chemistry per question. But even if you are not studying yet, even if you plan to only start studying after a few weeks or a couple of months, you can still attend these tutorials. We'll discuss some questions. We'll talk about some strategies. We'll get you in the mood for your studying, the questions are quite reasonable. And I'd also like to mention one thing, just in case any of you are looking at some of the questions and saying, my degree didn't include this or that or whatever. And in future tutorials, I want you to understand that working through the question is studying. So it's better to try to work through the question any way you possibly can during the tutorial, even if it means open book, even if it means you have to Google something or call a friend or call a professor. I don't think they will appreciate that on a Sunday, but even if whatever you can do to work out the question when you're given time during our sessions together, just do that rather than twiddle your thumbs and wait for the answers or just look at the back of the document, the PDF document for the answers. It's better to just work it through. It's really helpful. You see the answer choices, you have the uncertainty, and then you work it through, even if it's a topic that you've never seen before or you haven't studied before. Just go through it, and that way you're studying more. It's way more helpful than just seeing the letter or seeing the answer or whatever. Okay, so next time we're gonna start with question 6. We're going for all 100. And then when all those 100 questions are done, I'm sure with time, it may get a little bit more efficient. But when all the 100 questions are done, we'll just do DAT QR, so we'll go into quantitative reasoning. And if you want to joke, look at the back of your 2007 exam. It's so hilarious. The ADA does this every year. They misspell quantitative for quantitative reasoning. So albumin and quantitative, but it's okay. Most of the questions are great. Most of the questions are accurate for most of their exams. And so we'll get to all of that. I hope you found today's session helpful. And if there's any ways that we can improve it or make the teaching better or more background or whatever you're looking for, just post a comment, or you can always go to our website, which is datprep.com, and you can see that there, datprep.com. You can go there and you can leave a message. There's also email and so on and so forth. Oh, there's also a free exam. So I'll make sure that that's in the YouTube description. So datprep.com has a free practice test, and you get a scaled score, so you can use it like a diagnostic test or a prognostic test, depending on how you would like to use it. Okay, so that's enough jabber for today. I hope you found it helpful. I wish you good luck with your studies. Stay safe, and see you next week. Bye for now.